Oradan değiştirmek gerçek kamerada konuşmak bir mana olacak. Konuşmak bu. Canlı belki bir şey olacak. Kameradan çıktım ben.
Welcome everyone. Today our guest is Dr. Musa Sarabi. He has conducted his PhD studies at Koç University and Maxwell Institute for Intelligent Systems. His research focuses on nanotechnologies, that is microneedles and microfluidics. He will give you details. Thank you.
Thank you so much for the introduction. Hello, everyone, and thank you for taking time and coming. So today I'm going to talk about this topic, microneedles for drug delivery, and introducing engineering as for advanced delivery of medication to bodies. So first, I want to give a quick introduction of myself. So I have completed my Bachelor of Science in Primary Engineering at University of Taiwan, and from finishing PhD, I started my PhD in Biomedical Science and Engineering program at Koç University, and I was co-advised so my main supervisor was Professor Savaş Taşoğlu at Koç University, and my co-advisor was Metin Sepik from Max Planck Institute for Intelligent Systems. So it was a collaboration between these two universities, and so I completed my PhD mostly focusing on microneedles. So microneedles, as the name suggests, are micro-scale needles that are on an array or patch, and they can be used for a variety of biomedical applications. So throughout my PhD research, I conducted study on different types of microneedles, including hollow microneedles, which can be used for sampling body fluids, and also solid or dissolvable microneedles that can be used for delivery of drugs to the skin. So today, mainly I'm going to talk about this research, long-lasting simultaneous epidermal and dermal microneedle delivery. This was published in Advanced Materials Technologies a couple of months ago, and it is going to be featured on the cover of the magazine as well. So here, we focus on diseases of different depths. We have various diseases, including skin cancers, including different types of skin cancer like melanoma and basal cell sarcoma, or BCC, or squamous cell sarcoma, SCC, that happen at different layers of the skin. Or we can have skin inflammatory conditions such as psoriasis, that again can manifest at various layers of the skin, and bacterial infections, which also can happen in epidermis, dermis, or hypodermis layers of the skin. So the upper layer is epidermis, then we have dermis, and then we have the hypodermis in our like parts or floors of our skin tissue. So here, what we want to achieve is to address or access all these skin layers together. So we are introducing the non-linear design of microneedles on the array for this purpose. We are introducing two different designs. We are calling them the mountain design and the wave design. So Up until now, all the microneedle arrays had organization of needles with the same height or with the same diameter on the array. So here we're proposing this gradual increase of height from the sides to the central area. So in the central area, we have longer needles, and in side area, we have shorter needles. So it looks like a mountain from the side, as you can see here. So that is why we call it the mountain design. And we are introducing this one. So here we have two different base diameters for the needles. So one needle is partly larger, and then the other one is smaller, and then the pattern continues. And we're calling this wave design because of the similarity between these. So what can be achieved with these designs? So the mountain design, because it has these various types of the needles, can simultaneously access the skin layers. So here we can, as you can see, potentially we can address skin different layers, three of them at once. And what wave design affords us is that, so since the smaller needles will be biodegraded in a much faster kinetics than the larger needles, so these ones will take a little bit more time to biodegrade and to release the medication inside of them. So we can achieve multi-burst or long-lasting delivery of medications into our skin. So first, we did a simulation just to understand the feasibility of these designs, if they can be fabricated, and in which areas we need to be more careful while fabricating them. So the simulation results indicate that so the longer needles in the mountain microneedle are subject to be experiencing more stress. And here in the wave design, the needles with smaller diameters are subject to experience more stress. So these results indicate that we need to be more careful while we are demolding them or retreating them, especially in these areas. And also it also helped us to decide how small we can go for the wave design. So for fabrication, we used a combination of 3D printing, PDMS casting, and micromolding to achieve our microneedles. So first, we designed the master mold of these microneedles, and then these master molds are 3D printed using a stereolithographic technique, which gives us a very nice resolution. So over these master molds, we test PDMS, polymethyl. So it is a famous material to create molds. And then we achieve our fabrication molds after demolding. So the main material that we are using for fabrication of microneedles is polyvinyl alcohol. It is biocompatible, it's biodegradable, and it has the capability to encapsulate a lot of medications inside of it. So we pour the PVA solution into our molds, and upon two-step centrifuge and drying process and demolding, we finally can achieve our microneedles. And upon demolding, so the microneedles will look like this. This is the mountain design, and this is the wave design. And we can see that all the features that we wanted are very finely and precisely fabricated in a very nice way. Also, in order to further investigate these microstructures, we can do different types of microscopy. So here we can see SEMs of these needles as well. And we can see that not only they're fabricated successfully, but the precision of the technique is also really good as well. So, now we have fabricated the microneedles. We need to see if they are capable for drug delivery. We need to validate our theory. So, first step is the characterization of their drug delivery efficiency based on the degradation of the needles. So here you can see a time-lapse photos of the mountain design microneedle after being exposed to a solution that mimics human skin physiology in terms of pH. So the microneedle was exposed to this medium, and then it was washed with alcohol in each step, and then the microscopic photograph was taken over time until all of the needles were degraded inside this medium. By doing an analysis of the degradation of the needles, we are able to achieve these curves that give us some insights on the drug delivery efficiency of these microneedles. So here we can see the degradation of the needles for mountain design, and it shows smoothly how the needles are degraded into this medium. But more importantly here, for the weight design, because we are proposing the double dose delivery. So here we can see that the needles with a smaller diameter are degrading faster, and the needles with larger diameters are coming after them. So this gives us an insight that even we can further manipulate these parameters just to achieve or engineer how slow we want the second dosage to be delivered. Next, we did another technique for validation of the concept of drug delivery. So here we are characterizing their efficiency for model drug delivery. So we fabricated this custom setup that we filled in with bone gelatin that is famous for mimicking human skin tissue properties. We have a glass slide in one side, which gives us a very nice property in order to take a very nice microscopic photos, and then our microneedle was administered into this medium. And over time, as we can see in the next slide, we did time-lapse microscopy of the microneedles. So here, the microneedle is applied into the medium, and then over time, we can see how it is degraded, and it is delivering this blue dye as a model drug. So we can see over 85 minutes, almost like 100% of the array is blended into this medium. So we did some analysis on these data in order to achieve the depths and the weight of the delivered drug into the medium. So first, we can see the depths of penetration for the delivered model drug into the skin. So over time, of course, as it is also visible in the photos, the depths of penetration is increasing because the degradation is happening over time. But more importantly, we can also give an idea of the weight of the delivered medication over time as well. And we can see that 100% is really solving what happens over 85 minutes. So next, we are introducing this unique property for mountain design microneedle as well. So in fabrication, using some sticks and increasing the number of centrifugal steps into 4 instead of 2. We are able to encapsulate blue dye in the central area where the needles are longer as model drug number 1. And we are encapsulating the red dye in the side areas where the needles are shorter as model drug number 2. So what we are proposing with this technique is that not only we can achieve various layers of the skin at the same time, but also we can deliver 2 different medications into the administration site. This can be also very useful for skin cancer treatment, where we have different medications used for treatment of the tumor sites on our skin. So next, we are validating our microneedles on human skin as well. So what we do in these experiments is that we first used cadaver human skin samples from Koç University Hospital. So there are some surgeries that they have excess skin produced, so they kindly donated the skin for our research. First, we apply our microneedles into the skin samples. In order to standardize the tests, we are using the spray applicator, so people can argue, for example, someone has more force to apply the skin, or the degree that the microneedle is applied on the skin can be different. So in order to standardize, we use this spray applicator. And the microneedles were administered on the skin. So after administrating the microneedles, so the area that the microneedle was penetrated is frozen and embedded in paraffin wax, and then it is cut into very thin slices. And finally, by applying the HND staining and putting it under microscope, we are able to achieve these microscopic photos, which show the administration site of the needles on the skin. So here we can see a broader area that was exposed to microneedles, and we can see some close-up views of different sites. We can see the micro wounds and the micro punctures that has been created by the microneedles. So this shows the successful administration of microneedles on human skin cadaver. So this was a little bit more challenging to achieve. The human skin cadaver takes up itself and it dries. So in order to keep the skin fresh, we did a lot of efforts, like putting the PBS solution continuously on the skin sample, and also we used pins in order to open up the skin into the actual tension that it is experiencing while it is live on our skin. So when we could achieve some successful results here, we did experiment on microneedles on human skin in vivo as well. So our microneedle with methylene blue as novel drug, which is also harmless to humans as well, was administered on a human subject arm. So upon administrating, we can already see how the area looks, and we can see traces of the dye that has been delivered into the skin. So this part of the skin was taken out using biopsy from that human subject, and then we did the same steps that I explained for the previous slide to achieve these histological microscopic photos of microneedles. So again, here we can see the macro pores and macro punctures created by microneedles. And also we can see traces of the blue dye, as you can see here, and here, and here, and this one, and delivered into the skin that shows drug delivery capability of our microneedles. So from this research, what we can understand is that microneedles are creating novel paths for delivery of medications into our skin in a very painless manner. And also they propose a solution to needle phobia, and so it's quite nice. And also, so nowadays we are in the research, we want to focus more on sustainability as well. So compared to needle injections that we have stainless steel waste in microneedles, so it can be fabricated completely by degradable waste, so we have no waste or harmless waste. And finally, so these new engineering methods, especially nonlinear ways, introduce more personalized treatment procedures and can be applied for various biomedical applications. So thank you so much for your attention. If you have any questions, please feel free.
I have a question.
Yes.
In the slides, the stratum corneum seems to be ripped off in many cases. So do you have particular treatment for that, or is it just due to handling, tissue handling?
Sure. So let's show the slide. Previous one. So yeah. So the area that the microneedle is administered on our skin, usually it will heal itself within like almost a day. So there are these teeny tiny micro punctures that the body will take care of it.
How about the stratum corneum layer, which is pretty tough usually? So that layer doesn't exist in many of the places on.
So what we saw that almost in all cases, I also tried my microneedles on my knee. So it heals itself within 1 day. So this like wasn't much of a challenge for us. It is also has been reported that like microneedles heal, but not only heal, but also help with healing as well, like these dermatological microneedles, which is very different what we studied here, like these micro rolls that people do on skin. So it creates tiny micro punctures on your skin, but body is very able to take care of it.
Oh, I mean, it's the dead skin layer anyways, but it's typically very tough, so it may be not easy to puncture it without a metal needle.
So the aspect ratio of the microneedles are high, so they are able to puncture. So again, this isn't also challenging these studies. It's easily achieved.
Did you prepare the slides yourself? The histology.
Any other questions?
How bad does it feel? Does it hurt?
It doesn't hurt.
How about the sensation?
So when I was doing it on myself, well, of course, when I'm doing, and I am the one fabricating it, so I will say they don't hurt. But the human subject that we tried them on also didn't experience any pain. And also, there are studies specifically that have reported the pain experienced by microneedles is not like the case. Like people don't experience any pain.
How deep is it? How deep are the needles?
So they can be like in the. We can yes. Yes. So here we can see like they can go as deep as 1,200 micrometers. And also 800 for this case, so they are very thin, I think. Any other questions?
Since they have different lengths, how do you load the medicine first? Are you thinking about that? And you only performed the dye.
Yeah.
And when you use chemical, their diffusion, their interaction should be different. So how will you overcome that problem?
So in order to first encapsulate, I guess one more. Yes, here. Okay. So we directly add the dye or the agent into the solution here, and then we mix this really good. And by centrifuging, so all this matrix solution and the agent we can fills up all these cavities. So this is how we make sure that the dye or the agent is inside.
But it's only one agent, right? Because you said that since they are different, like you are suggesting some different
So yeah,
diseases, and you should use different chemicals then.
that is true. So first, so I want to say this is how we made sure that they're all in these cavities. For the case that we encapsulated a dye here and another dye in the side areas, so we covered these parts of the mold first, and then we did the central area, and then after drying, we covered this part, and then we did the centrifusion and filling for the area. So that is how also we achieved the multi-dye process. So as you said, different chemicals and different agents have different diffusion mechanisms into the skin. So that is also something that can be studied. However, so in the literature, the dyes that we have used are very famous for these studies, like rhodamine and tissue marking dyes. These are very well known for studying how they can act as a model drug, and then finally actual drugs can be used. So my other research, which hasn't been published yet, so in that case, we are actually loading the microneedles with the agent for BCC skin cancer. So we are using that agent, which is an actual drug, and in the same procedure, and we are not seeing any problems with the loading and the diffusion over time. So hopefully it will be published.
Yeah. What's the diffusion that?
So in that one, we are using 800 micrometer microneedles, so they're able to reach the 900.
Are they going deeper or just superficial stage?
So mostly we are aiming for that.
For certain layer.
There was a Metin blue picture, like figure
Yes.
that you can see, it can go a bit.
Yeah.
Right? Did
Yes.
you measure that?
Yes, in the next one, we can see the depths of the next one, please. So we can see the depths of penetration here.
Like 4 millimeters.
Yeah.
Future work, where would that go?
Okay.
How
So-
far would that go?
Yeah, so what I'm thinking for as the next step is to manipulate the structure of the microneedles based on what we need. So here we are showing that with these different sizes, you are able to achieve this simultaneous access or multi-burst delivery. So in a specific way, let's say that we have this skin cancer that needs the medication needs to be delivered every 4 weeks. So based on that need, we can design our wave microneedles in a way that the first series of the needles will be degraded over course of 4 weeks, and then the other one will be degraded in the course of 8 weeks. So we can achieve this multi-delivery based on for a real biomedical application. That can be a future work. The second one is combining these two techniques together. So not only we are using needles with different heights, but also we are manipulating their base diameter as well. So this can give us a very advanced technique that we are, for example, can we go to the first slide that we have the skin? Yes. Okay. So the first ones, we have the schematics of this, the first slides. Yeah, this can be good. Yeah. So by combining these techniques together, we can propose an application that we will say, for example, we are delivering this model drug number 1 into epidermis layer in the course of 2 weeks. We are delivering this model drug number 2 to hypodermis in the course of 3 weeks, and then we are delivering this model drug number 3 in the course of 4 weeks. So by combining these two techniques together, we can achieve that. So that can be a route for future works as well.
You also said this can be a good alternative for injection, right? We can needle. But also, as far as I know, there are topically applied creams for skin cancer or those kind of things. Are you going to compare this with the creams?
Yes.
Which one is better?
So in this unpublished work that I said, so we are encapsulating imiquimod in our microneedles for ECC skin cancer treatment. Imiquimod right now is treated in the form of topical cream for people who are experiencing this cancer. So in that work, specifically, we are comparing, we apply the same amount of medication in the topical cream on one side and the same amount of drug using microneedles. So topical creams, when we apply them, almost like 20 or 30% of it diffuses into our skin, and then the rest is usually, washed away or going into waste. So with microneedles, we are saying that we can increase that efficiency really in a very noticeable way. So that is one thing that we have worked on as well.
Do you think you can treat tumors that are close to the skin, like benign tumors also, weak bones and things like that?
So microneedles, so when they have access to this layer, they can release the drugs into our blood as well. So, and the blood circulates anywhere, so it can act as like any other kind of injection that the drug goes into our blood, and then the blood takes it all over our body. But since microneedles are applied onto skin, so skin diseases are most popular for microneedle studies. Because in case I have like a cancer in my intestines or GI tract, in that case, like eating pills that goes to that area or injections will be much of a better route than like applying onto our skin.
You have a slide about the mechanical properties. Can you go back to that?
I have only the simulation here. Next one. No. Yes.
Yeah, the one before, I think.
The one after this one. So these are the simulation results. We are giving the deformation and experience the stress on the microneedles and on the skin in the simulation.
So what's the stiffness like?
Stiffness of the microneedles, we haven't measured that. So it is made of PVA, so whatever is reported for PVA as polymer, it should be the same for microneedles.
During the application, though, you may have a different, adaptable, I don't know. So it's not just the material, but there's a layer which can bend, right? So
Yeah, the aspect ratio is important, and also so in order to standardize it, we use this spring applicator, so we took that out of the equation. So yeah. Sure.
I have a question about manufacturing. What kind of 3D printer did you use?
It was a stereolithography, SLA.
SLA. So you use
That works.
resin as a material, right?
Yes, yeah.
So after the printing, what kind of problems did you get? Because in the curing part and the needles are very, very small. Is there a challenge for you? And
Of course. So if we go to that next one, I guess. Yes, yeah. So here it's only a sub figure, but this is a very long story. As I said, these. So first, how to place them on the printing side is very important. So some printers are curing the material with special degrees, so that affects how the product is achieved. So first is placing the microneedles in the print area, and upon printing, so we need to do very like strong cleaning methods, like using sonication and washing with different alcohols in order to clean the residue and extra resin on top of the needles. Because if that extra resin is cured with the microneedles, then it is not a good product at all. So yeah, upon printing, so we wash a couple of times with different alcohols, we do the sonication, and finally curing with UV light the machine to finally achieve this. So these are the steps that we.
so in curing part, it will be a deformation, or the length of the needles can be changed, right?
This CatWorks 3D printer, it also cures, so we can manipulate how much it cures.
Okay.
So we are not curing it completely, like almost, it can stand by itself, so the force of gravity won't demolish it. So we cure it up until that point, so that is achieved experimentally. We used different exposures of the light sphere, and then we achieved that experimentally. And then upon washing, then we put it for final making it firm in the UV machine just to cure it.
So just one more question.
Yeah.
In Z plane,
Yeah.
the curing or printing is here, right?
Yeah.
Did you print it 90 degrees or what, just on the plate? Did you pre-print on the plate or make an angle?
So we like it almost 30 or 45 degrees.
Okay.
So it was helpful in achieving high resolution product. But 90 degrees worked much better as well. But in order to make it even better, so we used this 30 or 45 degree. So these are the microneedles. So it looks like it was. Yeah. Any more questions?
thank you.